In this lecture, I want to talk about the process of translation. That is the synthesis of a protein using information contained in a messenger RNA that ultimately came from the DNA. Now, again, the central dogma of molecular biology is that DNA makes RNA, makes protein, and we can see that in this figure with the DNA sequence shown at the bottom and a copy of the messenger RNA that's made from it above that. The information in the messenger RNA is translated by the ribosome. That is, the ribosome reads the messenger RNA and puts amino acids into a growing polypeptide chain according to the information that's contained in the messenger RNA. It knows the order to put things in by reading the sequence in the messenger RNA, and that sequence is read in 3 base units at a time. So those 3 base units have a name, and they're called codons. Now, I want to use that term and emphasize codon is a sequence that's found in a messenger RNA. I'll talk later about an anticodon, and anticodons are found in transfer RNAs. So codon is a sequence in a messenger RNA. It consists of 3 consecutive bases. This sequence shows the genetic code, and we can see the genetic code actually has that 3 base sequence that I talked about before. And the genetic code is interesting. If we take 3 bases and we have 4 possible bases for each position, that means that there are 64 possible codons, that is 3 base sequences of those 4 bases. Well, there's only 20 amino acids that are used commonly to make proteins. There's a 21st that is occasionally used, but for our purposes, it's not really relevant here. Well, if we have 20 amino acids used to make proteins and we have 64 possible codons, either we have to not use some of those codons or we have to use some of those codons multiple times for a given amino acid. Well, it turns out that that's the most common thing that isn't in fact what's done. That is that multiple codons are used to encode for a given amino acid. So we can see, for example, leucine is an amino acid that is used by several different to be coded for by several different codons. What this means then is if mutation happens, some simple mutations that can occur have no effect on which amino acid is put into a protein, whereas other mutations may have big effects on that. When we look at the genetic code, there's a couple of things that I want you to note. You're not going to be responsible for knowing any of the sequences except the following ones that I'll describe to you here. First of all, you should know that all proteins start with the same codon, that is AUG. Okay? That AUG specifies methionine. Now there are a couple of rare exceptions to that, but for our purposes we're going to say that AUG is the start codon, and since AUG codes for methionine, only methionine is used to start proteins, and AUG is the start codon. I'll call it that commonly, okay? So the start codon is one of the codons that you should know. There are also 3 codons that have the function of telling the ribosome when it's translating the messenger RNA sequence. 3 codons have the function of being what's called the stop codon, meaning that they're telling the ribosome, okay, we're done. This is the place where you should stop making the protein and let everything go. Those 3 stop codons are as follows: UAG, UGA, and UAA. Those 3 are the stop codons, and those are the only ones in addition to the AUG for which you should know the sequence and the function. Now, if you look at this genetic code table, you'll notice something interesting. That if we look at for some of these amino acids, we'll see that the entirety of the sequence is redundant. Let's take a look, for example, at leucine. Leucine is an example of an amino acid that is specified by a redundant code. A redundant code meaning multiple codons all can specify leucine. Leucine can be specified by CUA, CUG, CUU or CUC. In other words, it doesn't matter after the CU what nucleotide is present. It still specifies leucine. And we see this is very common for several of the amino acids in the genetic code. It means that that third base of the genetic code is what we call a wobble position, meaning that it can vary, but it frequently doesn't affect which amino acid it specifies. The first two are the most important two nucleotides of the genetic code in specifying amino acids. The third base is at the 3' end of the codon. Notice that the codon goes from 5' to 3'. So a CUA would have the C at the 5' end, okay, and the A at the 3' end of that codon. An anticodon is anti-parallel to that. The anti-parallel nature of that means that the wobble position of the anticodon is not going to be at the 3' end of the anticodon, it's going to be at the 5' end of the anticodon, because the 5' end of the anticodon forms base pairs with the 3' end of the codon. So the anticodon has the wobble base complementary to the 3' end of the codon, which puts it at the 5' end of the anticodon. Okay, well, if we look now at the structure of the transfer RNAs, we see that the information necessary for the anticodon, which is being read by the ribosome, to be read, is very far away from the 3' end of the transfer RNA. Well, why is that important? Well, it turns out that there's an enzyme that has to put the amino acid onto the 3' end of the transfer RNA. But in order for it to put the amino acid on here, the proper amino acid on here, it must also read this, because this anticodon is going to be used to tell the ribosome which amino acid is attached, and if the wrong amino acid gets attached based on the anticodon, then translation won't occur properly. Well, there are enzymes that do this function, that is, they read the anticodon and then put the proper amino acid at the 3' end of the transfer RNA. And these amino acids are called amino-acyl tRNA synthetases. Now that's a mouthful of a name, and in the class I usually joke with my class and we give it a simple name, and you're welcome to use the simple name that I use in class instead of an amino-acyl tRNA synthetase. But suffice it to say that those enzymes are responsible for making sure that the amino acid is properly attached at the 3' end of a transfer RNA. This process is called charging of a transfer RNA. And some people call it the first step in the translation process, okay? So you can do that if you wish. What I refer to in the translation process is initiation, elongation, and termination, kind of like we saw in transcription. But if you want to consider this a first step, that's also fine to do. Now, that is a very important step in that process because, again, we have to have the right amino acid at the 3' end that corresponds to the anticodon down here. Remember that the anticodon is going to form base pair with the codon in the messenger RNA in the ribosome during the translation process. Okay, well, during the translation process, there is, in fact, a ribosome that comes together. And as I talk about in class, I won't go into all the details here, there are two components of a ribosome, a large subunit and a small subunit. And we're going to see how they come together in the initiation process in just a minute. Suffice it to say that when they come together, they create a set of 3 structures that are important for us to understand in the translation process. And those 3 structures are known as the A site, the P site, and the E site. And these 3 structures are formed as a result of the joining of the large subunit and the small subunit of the ribosome. And by the way, when the ribosome forms that structure, the messenger RNA is going between those subunits. So that's what's there. The formation of this structure is what gives rise to the A site, the P site, and the E site. Well, what are they? Those A site and P site and E sites are places where the transfer RNA comes in during the translation process. And that coming in turns out to be important, as we'll see, as the ribosome is moving along that messenger RNA. Now, one of the things that happens in translation is that the ribosome has got to know, well, where is the AUG, that start codon, where is that AUG that I know where to start the process? In eukaryotic cells, most eukaryotic cells start with the first AUG that the ribosome encounters. Not all, but most start with the first AUG that they encounter. Prokaryotic cells don't have that property. Prokaryotic cells may have other sequences or other AUGs before the first one, and if the ribosome started with the wrong AUG, there would be problems. Further, in prokaryotic cells, the messenger RNAs that are made in the prokaryotic cells have multiple genes within them. So the prokaryotic ribosome has to be able to recognize several different start codons. So how does it know where or what a given AUG is a start codon? Well, it turns out that prokaryotic cells have contained within their messenger RNA a sequence called the Shine-Delgarno sequence. Now, the Shine-Delgarno sequence is a sequence that is complementary to a portion of the 16S ribosomal RNA. Now, I mentioned this in class, but I'm going to repeat it here. The Shine-Delgarno sequence is complementary to a sequence in the 16S ribosomal RNA. The 16S ribosomal RNA is a component of the small subunit of the ribosome. So the ribosome is carrying this 16S ribosomal RNA, and when the messenger RNA combines with it, they form base pairs. And they form base pairs such that the AUG is properly lined up to be in the position to start translation. And we'll see that during the initiation phase. But I want you to remember that Shine-Delgarno sequence. I'll repeat it again. The Shine-Delgarno sequence is a sequence in the messenger RNA that's complementary to the sequence in the 16S ribosomal RNA, and that Shine-Delgarno sequence is about 10 nucleotides away from the AUG. So once all that information comes together, the ribosome knows where to start the process of translation. Now, one last thing I want to say about prokaryotic translation. And by the way, there are some differences between prokaryotic and eukaryotic translation, and I will only talk about those differences that are significant. There are different names of proteins, and the eukaryotic process is somewhat more complicated, but the overall general process is the same. So only where there are differences will I really distinguish those. One of those differences occurs in prokaryotic translation, and that occurs with the very first amino acid that gets put into a prokaryotic protein. It turns out that the very first amino acid that's put into a prokaryotic protein is methionine, but it's a chemically modified form of methionine known as formylated methionine. Now, I mentioned this in the lecture as well, but formylation involves putting a formyl group onto methionine to basically chemically protect it from reacting with itself, and I won't go into that here. That's discussed in the lecture. But that formyl methionine is the first amino acid in a prokaryotic protein being made. That is not found in eukaryotes. Eukaryotes don't need to do that for various reasons, as mentioned in the lecture. Okay. Now, once so that formyl methionine called FMET is an important consideration in the initiation of protein synthesis. Now, the initiation phase, as I mentioned earlier, protein synthesis, or the process of translation, occurs in three general steps: initiation, elongation, and termination. So in the initiation phase, what the cell is doing is the cell is constructing a ribosome on a messenger RNA. So let's talk about that process. In the construction phase, during the initiation, there's several things that have to happen. The first subunit of the ribosome that binds is the small subunit of the ribosome. Now remember, it's the small subunit that contains the 16S ribosomal RNA, and that 16S ribosomal RNA is complementary to the Shine-Delgarno sequence. And that Shine-Delgarno sequence is helping to align the proper place for the AUG for the translation process. So in the 16S ribosomal when the small subunit that contains the 16S ribosomal RNA binds to the messenger RNA, it scoots along looking for that complementary Shine-Delgarno sequence, and when it finds it, it stops because it knows that the start codon, that is the AUG, is very close. There are some proteins called initiation factors, or IFs, that help this process to occur, and I don't hold you responsible for those in this class. After the Shine-Delgarno sequence has formed its pair with the 16S ribosomal RNA of the small subunit is positioned in the right place for translation to start. After that occurs, the large subunit can then come along and form that snowball or snowman-like structure that we see associated with a ribosome. At this point, the ribosome has a, I'm sorry, I'm screwing up here. I just can't see it. Okay. Let's start that over. Let me think about that. Okay, all right. In the initiation phase of translation in prokaryotic cells, and by the way, in prokaryotic cells, initiation is very much like it is in eukaryotic cells, so I won't really distinguish that overall process here. I noted earlier, of course, that there's an FMET used in prokaryotic cells, but not in eukaryotic cells. But other than that, for our purposes, we'll consider them essentially the same. In the initiation phase of translation, the small subunit, which contains that 16S ribosomal RNA, is binding to the messenger RNA. That 16S subunit slides along the messenger RNA until those complementary sequences between the Shine-Delgarno and the messenger RNA and the 16S ribosomal RNA in the small subunit align. When they align, the small subunit is properly positioned for the translation process to begin to occur. Now, that binding of the small subunit at the proper place is really the first step in the process of translation. There are some proteins called initiation factors that help facilitate all of these steps in initiation. They're called IFs, and there are 3 of them, but you're not responsible for knowing which one does what. After the small subunit has bound to the messenger RNA, then it's time for the amino acid FMET, which is linked to a transfer RNA that can come in and bind to this complex that's starting to be formed. So the complex contains the small subunit of the ribosome, the messenger RNA, and what will become the P site. Remember I talked about the A site, the P site, and the E site. Well, the place where the transfer RNA that contains the FMET binds is what will become the P site. What is the transfer RNA doing? The transfer RNA is forming a base pair between the anticodon of the transfer RNA. Remember, the anticodon's at one end, the FMET is up at the other end of the transfer RNA. There's a base pairing between the anticodon of the transfer RNA and the AUG codon of the messenger RNA. And this is now positioning that FMET transfer RNA in what will become the P site. Well, after that proper FMET transfer RNA has come and bound, then the large subunit of the messenger RNA can bind and create that familiar snowball-like structure that we see associated with a ribosome on a messenger RNA. Okay? At this point, the next transfer RNA can come in because remember we're going to make a polypeptide chain, and each amino acid will be carried to the ribosome during the process of translation, the ribosome has assembled at this point. We've got the small subunit, we've got the large subunit. We have the FMET tRNA in the P site, and now we have this other site in the ribosome that's open. We're getting ready to do the process of elongation. In the process of elongation, the first thing that happens is a transfer RNA carrying the next amino acid comes in. Well, what happens with this? Well, there's a couple of considerations. First of all, the A site of this ribosomal complex is where the new transfer RNA will come in. Well, remember, the cell has got a lot of transfer RNAs that come in. Not all of those are going to be the right one as the next one. What is the right one that's the next one? Well, the right one that's the next one is the one that is going to have an anticodon complementary to the second codon of the messenger RNA. Remember, the first codon has this base pair between the AUG of the start codon and the complement to that on the FMET tRNA. So the next codon is the second one. The cell needs to have, or the ribosome needs to have, a transfer RNA that's complementary to that second codon. Well, if a transfer RNA comes in that has an anticodon that is not complementary, it gets removed. Well, how does it get removed? It turns out that when transfer RNAs are moving around inside of a cell, they are, I won't say attached to, but they're bound by a protein called EFTU. Now, EF stands for elongation factor, and TU stands for TU, okay? So elongation factor TU, I mentioned in class, holds tightly onto the transfer RNA, and it covers up the amino acid so that the bond between the transfer RNA and the amino acid can't be broken by water, and I talk about that in the class lecture. Well, EFTU protects that bond, but it also does something else. When the transfer RNA comes into the A site, EFTU looks to see if, in fact, this is the right transfer RNA, and it answers that question by looking to see, do proper base pairs form between the anticodon and the codon that's in the A site? If they aren't proper, then EFTU is what's responsible for taking that amino acid away, okay? On the other hand, if proper base pairs are formed between the anticodon of the transfer RNA and this codon that's in the A site of the ribosome, then EFTU says everything is fine. It cleaves GTP and then it leaves that transfer RNA in the A site of the ribosome, okay? Well, the next step in the process of elongation means that that amino acid that's on that transfer RNA in the A site has got to be joined to the FMET that's on the transfer RNA in the P site, okay? So that involves joining those two amino acids together to form a peptide bond. Well, peptide bonds are formed inside of ribosomes as a result of action of a ribozyme, okay? A ribozyme I've talked elsewhere in class. Ribozymes are catalytic RNAs, okay? It's not an enzyme, it's a ribozyme in a ribosome, okay? Don't get confused with the names there. A ribozyme catalyzes the formation of a peptide bond between those two amino acids, between the amino acid on the P site and the amino acid in the incoming amino acid in the A site. A peptide bond joins them together. They are left linked to the tRNA in the A site. The tRNA that was in the P site now slides out, okay? We're doing the process of elongation. Well, how does it slide out? It slides out because the ribosome itself slides down. And how does the ribosome slide down? Well, that's as part of a process we call translocation. And there's another protein that facilitates that. That protein is also an elongation factor. It's called EFG. And EFG catalyzes the movement of the ribosome by 3 bases, 1 codon, along the messenger RNA, going from the 5' direction towards the 3' direction. Notice that all these processes, replication, transcription, translation, are all occurring 5' to 3'. Okay? So the EFG is catalyzing the movement of the ribosome in the 3' direction, 3 nucleotides away. And now that amino acid that was in the P site has now moved to the E site. It's getting ready to exit. That's what the E stands for. Okay? The amino acid that was on the transfer RNA in the A site is now on the transfer RNA that's in the P site because it has moved 3. And the A site is left open, meaning that a new transfer RNA can come into that A site. And when it comes into that A site, this process of elongation just continues on and on and on. Peptide bonds are being formed continually so long as the proper tRNA is being brought into the A site. When the improper one is brought, then it just gets kicked out until the proper one gets brought in. Well, this elongation process occurs repetitively until the ribosome has to stop. And how does the ribosome know when it has to stop? It has to stop when it encounters a stop codon. Now, a stop codon, I told you before, there are 3 different stop codons that are found in the genetic code: UAA, UGA, UAG. If any of those appears in the A site of the ribosome knows it's time to stop. At that point, there are proteins called release factors that will come into the A site, and they will do 2 things. One is interesting. They will carry a molecule of water, and they will release it to the tRNA polypeptide chain bond and break it. That releases the polypeptide that was being made, that was being carried on that tRNA to be released into the cell. And then the second thing that the release factor will do is it will favor the breakdown of that complex of the ribosome. So the small subunit, the large subunit, and the messenger RNA will all be released, and the translation process at that point is complete.